Okay, so it's time for us to talk about how a cell prepares to divide. And the biggest thing that a cell has to do before it divides is it has to copy its DNA. And that process is called DNA replication. This happens during the synthesis phase of the cell cycle. We call DNA replication semi-conservative. And what that means is that we keep half of the original strand of DNA in each new molecule. So the DNA is going to separate. We often call this unzipping. And then we are going to use the original strands as a template. And those templates are then going to be used to build the other side of the DNA molecule directly on the original strand, so that when we get our final product, we will have two strands that are identical to each other, where half of each strand, one backbone essentially, was the original DNA, and then the other backbone is newly built. There are three main stages of replication. There's the unwinding phase, the base pair phase, and then the joining phase. So unwinding, we have three steps. The enzyme called helicase comes in, it unwinds and unzips the DNA. When we unzip the DNA, what we're meaning by that is that we're breaking the hydrogen bonds that hold together those base pairs. So we're coming through and we're unraveling or unwinding that double helix. So we're taking the twists out of it. And then we're going to break those hydrogen bonds so that we can pull the two sides apart. And now that the DNA is open, we're going to hold it open with SSBs, which are single-stranded binding proteins. And those are going to basically prevent the DNA from reforming those hydrogen bonds. Then we're going to come through and we're going to put on an RNA primer, and that is done by RNA primase, which adds just a primer is just a short segment of RNA that's built onto the DNA molecule. Okay, so here's what that looks like. Okay, so we have our DNA here, right? And we're coming through and we're unzipping it. So that's what helicase is doing. It's going through and it's breaking those hydrogen bonds. And once they're broken, we're laying down an RNA primer and we're putting down some single-stranded binding proteins to prevent those hydrogen bonds from reforming. The primer is going to be important in the next part of this process, about joining together the next parts. So that's going to show up again. Okay, now the base pair rules. So the enzyme DNA polymerase is going to come through and it's going to match the nucleotides that are on the strand with a new nucleotide. So this is where the base pair rules really come into play. Remember, A always bonds with T, C always bonds with G. So if we have an A on our original strand, DNA polymerase is going to bring in a T to match up with it. DNA polymerase can only work in one direction. So it has to work in the 5' to 3' direction. If you'll remember the coloring page that we did in class, that was where the letters, the way the carbons are arranged. So on the deoxyribose sugar. So those can only work in one direction. Then we have, because of that, we have some problems that arise. Because one works in the opposite direction, or one strand is running opposite to the other, we have one strand that's running in the correct way for DNA polymerase to work. We have a second strand that's running in the opposite direction. And so we can build that strand that's running in the correct direction in one continuous piece. But the other side that's running in the opposite direction, basically we have to build segments of it in the 5' to 3' direction and then connect them later. Those segments are called Okazaki fragments, after the scientists who discovered them and figured out this process. And those fragments are then pieced together by a different enzyme called DNA ligase. Okay, so let's kind of talk about what 5' to 3' is. So 5' is the carbon atom that attaches to the phosphate group, and then we have the 3' here that is attached to an oxygen. And so we can kind of figure out which direction it's running based on how we kind of count from this oxygen. So we go around like this. Here's the 3' going back towards this phosphate group, and then we have the 5' carbon right here. And you can see that the oxygen is pointing in this direction. That kind of can help you figure it out too. And then the oxygen is pointing in that direction down on this side. Okay, so here's what the actual building process looks like. So we have DNA polymerase on our leading strand here, running 5' to 3', and it is just building right along. This is why it can build a continuous one, because this strand is running in the proper direction for this enzyme. But over here, you can see there's a lot more going on. So we have our RNA primers here. Those primers are going to be where we begin to build our fragment. So we're going to attach at the primer, and then the DNA polymerase is going to build in the direction it needs to from that primer until it reaches another primer. Then we'll remove the primer and we'll link together the fragments with DNA ligase down here. You can see that's what it's doing. So if you need kind of help figuring out what's going on here, these tags on this can be very helpful to kind of figure that out. Okay, joining. So now we need to make sure that all of the backbones are tied together and that everything is linked the way it should be. That's what we're going to do during the joining process. So when we come into contact with an RNA primer, we're going to remove that primer and fill it in with DNA nucleotides. And then once it's filled with DNA nucleotides, then the whole backbone can be joined together and link the sections that we built. Now, DNA does not happen just one way to the other. So it's not like we start at one end and then work our way to the other end. We actually form replication bubbles. That means that we have enzymes working in both directions in multiple different places along the DNA strand. And as they kind of work out from the center of their bubble, they link up and they become much larger. This allows DNA replication to be much faster. It allows it to occur in a short period of time, and it allows it to be a little bit more efficient. Okay, so just to remind you what we're talking about when we talk about semi-conservative. So if we have our original strand of DNA here, we unzip it, we bring in these new nucleotides here, then you can see how our exact copies now are each strand has an original and a new strand after the replication process. But definitely you should be able to identify what's wrong with this picture at this point. All right, that will do for DNA replication. This was just a quick run through. We'll probably be talking more about this in class as well.